Welcome to this explainer. Today we're jumping right into the rigorous, absolutely fascinating science of the endogenous modulation of GLP-1 via plant protein consumption. Essentially, we're talking about using specific foods to naturally stimulate your body's very own metabolic hormones. Right now, we're witnessing a massive paradigm shift in how we understand metabolic health. The data clearly shows us that dietary proteins aren't just passive building blocks for your muscles. No, they're actually dynamic, highly bioactive, signaling molecules that are literally capable of commanding our entire endocrine system.
To comprehensively cover everything today, we're following a highly structured path. We'll start with the enteroendocrine secretagogue axis, move into molecular mechanisms of receptor activation, explore microbiome dynamics, compare the efficacy of different botanical sources, break down strategic optimization and dosing, and finally, look at the real-world metabolic consequences and clinical outcomes.
Okay, Section 1, the enteroendocrine secretagogic axis.
We have to begin at the absolute foundation of gut-derived metabolic regulation. As you can see, clearly defined on screen, the crucial players here are your L cells. These act as your gut's native intelligence network. Anatomically, they're distributed all throughout your intestinal tract and are equipped with microvilli that extend right into your digestive system. Think of them as tiny antennas that are constantly sampling the food you digest. They natively sense the nutrients you eat and in response release that master satiety hormone we've all heard about, GLP-1. This single hormone is the cornerstone of metabolic regulation. It dictates insulin secretion, severely slows stomach emptying, and signals profound fullness directly to your brain.
Now, historically, modern medicine has tackled this axis purely through pharmacology. And look, while synthetic therapies do provide massive GLP-1 stimulation that's highly effective for weight loss, they often carry huge financial costs and severe gastrointestinal adverse effects, things like intense nausea and uncomfortably delayed stomach emptying. This reality has prompted a massive scientific imperative to find non-pharmacological dietary interventions. The big question we're answering today is, can we stimulate the endogenous secretion of GLP-1 directly from a patient's own native L cells? Let's move to Section 2, Molecular Mechanisms of Receptor Activation.
Now, what's really interesting here is how your body physically senses these plant proteins at a microscopic cellular level. If you just eat a raw, intact plant protein, those macromolecules are simply too large to directly bind to your L cell receptors. They've got to be broken down. And as the label on screen emphasizes, this makes hydrolysis absolutely critical. By enzymatically predigesting the protein into a hydrolysate, specifically breaking it into tiny fractions small enough to completely bypass the slow, rate-limiting process of stomach digestion, we allow these highly bioactive peptides to rapidly flood into the small intestine. This step-by-step breakdown brilliantly illustrates the acute biological journey. Step 1 is that crucial gastric hydrolysis we just talked about. In Step 2, these tiny botanical peptides are physically shuttled into the L cell. This immediately triggers Step 3, a rapid electrical shift that floods the cell with calcium. And finally, in Step 4, this massive calcium influx acts as the physical trigger, literally squeezing the GLP-1 hormone out of the cell and straight into your systemic bloodstream.
But here's the thing, this cellular transport doesn't just work in isolation. It operates in tandem with the calcium-sensing receptor, or CSR. Specific peptide sequences, like the ones rigorously identified in pea-protein hydrolysates, lock directly into the venous flytrap domain of this receptor. I just love that vivid biological imagery. This exact binding amplifies the secretion signal, triggering a complex chain reaction that ensures a really robust, sustained release of GLP-1, all without exhausting your native endocrine system.
Additionally, as we've highlighted here, basic L-amino acids from plants activate a secondary sensor called GPRC6A. Activating this specific sensor triggers the CAM-KI pathway, which mobilizes GLP-1 vesicles for immediate release. This is phenomenal because it essentially proves our gut has naturally evolved to seamlessly read specific botanical blueprints to regulate our overall metabolic tone.
Let's dive into Section 3, microbiome dynamics and secondary pathways.
You see, protein digestion is completely intertwined with microbial influence in your lower gut. A really common misconception is that protein sensing only happens way up top. While the small intestine absolutely handles the acute 60-minute post-meal hormone spike via those direct sensing pathways we just covered, the colon actually contains the highest absolute density of L cells in your body. The colon is responsible for sustained long-term GLP-1 release, and this is orchestrated entirely by your gut microbiota.
The gut microbiota acts as a literal master switch here.
When intact botanical fibers and escaping plant proteins finally reach the colon, bacteria ferment them into short-chain fatty acids, which aggressively upregulate GLP-1. Even more fascinating, the regular consumption of specific plant proteins actually alters your bile acid pool. Your microbiota converts primary bile acids into secondary bile acids. These then act as high-affinity triggers for the TGR5 receptor on your colonic L cells, generating a profound, heavily sustained GLP-1 secretion.
Section 4. Comparative efficacy of botanical sources.
Let's examine the actual clinical data, because the ability to stimulate GLP-1 is absolutely not shared equally among all plant proteins. If you look at the breakdown we've aggressively labeled in this table, the differences are stark. P hydrolysates are the best supported, vigorously locking right into our receptors. Rice hydrolysates present a uniquely powerful dual action. They stimulate secretion while simultaneously blocking the enzyme that naturally destroys GLP-1 in your blood. Soy peptides speed up stomach delivery for a preemptive insulin spike, and interestingly, potato isolate strictly dominates a completely different satiety pathway called CCK, but entirely fails to show reliable GLP-1 induction.
It's also absolutely crucial we impartially look at the data on wheat proteins. There's a widely circulated hypothesis out there that gluten digestion yields opioid-like exorphins that unnaturally stimulate appetite and drive weight gain. Well, rigorous endocrinological studies have definitively debunked this. The clinical reality, as shown on screen, is that these peptides do not cause metabolic slowing. Instead, low molecular weight gluten fractions act as highly potent anorexigenic agents that aggressively suppress food intake and actually lower overall blood glucose.
Moving on to Section 5, Strategic Optimization and Dosing.
Translating all these biochemical pathways into actionable clinical reality requires incredibly strict optimization. You really can't just casually drink a standard plant protein shake and expect miracles. First, let's talk total daily volume. To prevent the severe lean muscle degradation that's so frequently seen in rapid weight loss or concurrent pharmaceutical GLP-1 therapies, your daily plant protein intake must explicitly hit a specific threshold between 1.0 and 1.6 grams of protein per kilogram of body weight per day.
To actually trigger those cellular receptors, you've got to divide that intake into 25 to 40 gram increments per meal. Front-load your breakfast with a hydrolate to establish a really strong appetite-suppressing baseline for the day. You want to preload it 15 to 30 minutes before eating carbohydrates to leverage the glucose-dependent insulin response. And crucially, notice this on the list, you must strictly avoid heavy seed oil emulsifiers. Combining slow-digesting industrial fats with a GLP-1-induced delayed stomach emptying will literally lead to severe bloating and nausea. Keep it clean and make sure to combine it with intact dietary fibers to feed that secondary colonic pathway we just covered.
Finally, Section 6. Metabolic consequences and clinical outcomes.
So what actually happens to human metabolism when we execute this protocol perfectly? The systemic impact is absolutely profound. Locally, in the gut, the ileal brake effect violently slows your gastrointestinal motility. This severely flattens the systemic absorption curve of all nutrients, protecting your peripheral tissues from dangerous spikes in blood glucose and ectopic fat deposition. Neurologically, circulating GLP-1 and PYY cross the blood-brain barrier to forcefully suppress hunger-driving neurons. This leaves the POMC pathway completely uninhibited, triggering a deep, undeniable sense of neurological satiation. And, perhaps most importantly, by supplying highly bioavailable targeted doses of protein, we mechanically activate the MTR pathway. Optimized plant proteins effectively resolve the physiological paradox of rapid weight loss. They ensure your body aggressively burns adipose tissue, fat, while meticulously preserving your vital, highly metabolically active skeletal muscle. As we witness this massive, completely unprecedented shift in endocrinology and nutrition, one really has to wonder, are these highly bioactive botanical signaling molecules the holistic key to mastering our metabolic future? Could precision-dosed plant protein hydrolysates actually become the ultimate universally accessible foundation for combating global metabolic dysfunction? It's a truly fascinating paradigm shift, and the clinical data strongly suggest we are just scratching the surface here. Thank you for joining me on this explainer. Keep learning, and I'll see you in the next one.